Welcome, everyone. We're delighted to have you join us today for the webinar from Dyne Therapeutics, who provide an update on the DELIVER trial of Dyne 251. This is the company's registrational trial in Duchenne patients with mutations amenable to skipping exon 51. This is an exciting time as the company shares this new long-term clinical data. I'm Michael Kelly. I'm the Chief Scientific Officer at Cure Duchenne, and I'm going to get us started with a bit of housekeeping. We'll have an opportunity for Q&A today. You can click on the Q&A button at the bottom of the screen and submit a question anytime throughout today's presentation. There will be a moderated Q&A session at the end of the webinar, but we may not have time to answer all of your questions live. For those left unanswered, we'll work with the Dyne team to follow up with you after this webinar. After today, the webinar will be available on Cure Duchenne's website as a resource for the community in case you would like to share it with others. So let me begin. Dyne has developed the FORCE platform to efficiently deliver therapeutic cargo in a specific targeted manner to muscle and to the central nervous system. This is powerful technology that allows the company to target the genetic cause of a disease. To talk more about the platform and provide an update on the results of the DELIVER trial of Dyne 251, we've Dr. Ashish Dugar, the Chief Medical Affairs Officer at Dyne, and Catherine Beaverson, the Vice President of Global Patient Advocacy. So without further ado, let me hand it over to Ashish. Take it over from here.
Great. Thank you, Mike. So first, I'd like to thank Deborah, Mike, Liana, Chelsea, and the entire Cure Duchenne organization to present our most recent data that we released in March at the MDA annual conference. As always, it's a privilege to speak to the Duchenne community, and I want to thank you for your time and attention today. And as Mike said, with us today is also someone you know very, very well, has been in the community for a very long time, our Head of Patient Affairs, Catherine Beaverson. So the two of us will be going through this and answering questions at the end of the presentation. I'd like to remind everyone that we'll be making forward-looking statements today, and you can refer to our website for more information. Next slide, please. So I always start with this slide. You've seen me present this before, and it's really important. At Dyne, it all starts with our mission: to deliver life-transforming therapies for people living with serious muscle diseases. Our mission is woven deeply into our core values, and it inspires our employees each and every day. Next slide, please. So I'll talk a little bit about the FORCE platform. This is a different slide than you've seen me present in the past. So I'll just hit some highlights. As you know, the FORCE platform was developed to overcome limitations of oligonucleotide delivery, such as exon skipping PMOs, to muscle for the treatment of rare neuromuscular conditions. Now, the platform consists of an antigen binding fragment or an antibody fragment or a Fab that is specific and binds tightly to the transferrin receptor or TFR1 receptor. And you can see sort of in the middle there, that Y-shaped figure is a monoclonal antibody, and our Fab is that top portion of the monoclonal antibody. And we use the transferrin receptor because it allows for broad distribution of disease-modifying payloads to tissues that are relevant to the disease pathology. Now, the choice we made to use a part of that full antibody to the TFR1 receptor is really important, as it does not include elements that may lead to greater immunogenicity, and also the Fab's smaller size makes it easier to get into muscle cells. Now, the Fab is linked to a payload, and the modularity of this FORCE platform really provides the flexibility to send different types of payloads that are rationally selected to target the genetic basis of the disease that we're trying to attack. So, for example, FORCE can deliver antisense oligo, or ASO, payloads, such as exon skipping PMOs, that need to go to the nucleus of the cell for conditions such as myotonic dystrophy type 1 and Duchenne. Alternatively, it can deliver other materials, such as something called siRNA payloads, if you want to attack targets that sit in the cytoplasm for conditions like FSHD. And ultimately, we can deliver these payloads to the cellular compartments where they can most effectively target the genetic basis of disease. Next slide, please. And the FORCE platform was designed to overcome one of the greatest barriers in effective treatment, which is to be able to efficiently deliver the ASO to where it needs to go while minimizing off-target delivery to other organs or tissues. So the choice of the Fab targeting TFR1 allows the FORCE platform to leverage the body's natural cell biology. The transferrin receptor 1 is highly expressed in the body, including on the surface of skeletal muscles, including breathing muscles, cardiac, as well as smooth muscle cells, which enables the targeted delivery of therapeutics to these tissues because they're really important in terms of the disease pathology. And Dyne 251 is designed to leverage the TFR1 to deliver exon 51 skipping PMOs to the nucleus of cells in the affected tissues, such as skeletal muscle, including diaphragm, and cardiac muscle, to ultimately increase the production of dystrophin. And the goal is to make a shortened functional version of dystrophin throughout the body. Next slide, please. So in a mouse model of Duchenne, this is a very common mouse model, the MDX mouse. The FORCE platform has demonstrated broad delivery of the PMO to cardiac and skeletal muscle, including the diaphragm, with significantly higher exposure levels, the ability to really deliver much more than what we call naked or unconjugated PMO. The broad distribution allowed for robust and sustained or durable restoration of dystrophin expression, which you can see in the green staining on the top here, and also restored muscle architecture, which you can see in the red staining. Now, in the clinic, we can only explore the ability to deliver PMO to skeletal muscle following Dyne 251. And in the next section, I will show data that support distribution of skeletal muscle that is translating to early and sustained improvement in functional outcomes in individuals enrolled in the DELIVER clinical trial. Now, distribution to additional muscle types, such as cardiac and diaphragm, can be studied by looking at cardiac and pulmonary endpoints in the trial, which we do have. But this data will likely take longer to emerge. Next slide, please. So in this first section, just a few key points to take away. Really, as I said, the FORCE platform was developed to overcome the limitations of delivering these ASOs to muscle where they need to go for these rare neuromuscular diseases. As I mentioned, the platform uses the natural expression of the TFR1 receptor in muscle, as well as CNS, because the TFR1 is also located on the blood-brain barrier. And this allows targeted delivery of a rationally selected payload with the goal of addressing the underlying cause of disease. And we've had preclinical data that's been published that has demonstrated that by leveraging the TFR1, the FORCE platform achieves broad distribution to tissues that are relevant in the disease pathology, including skeletal muscle, respiratory muscles, cardiac and smooth muscles, as well as in the central nervous system. Next slide, please. So I'm going to transition to some of the clinical data now, and just very briefly spend a few minutes on DMD, which you all know very well. As you know, Duchenne is caused by mutations in the DMD gene, which results in the inability to make functional dystrophin protein. Now, PMO-induced exon skipping can restore that open reading frame of the DMD transcript and enable the translation of an internally yet functional form of dystrophin. And I know that this audience understands this very, very well. Next slide, please. So when it comes to therapeutic approaches in Duchenne, there are three key considerations that we believe are important to keep in mind for any therapeutic. First, increased dystrophin production, the production of high-quality functional dystrophin, and then the broad distribution of the exon skipping PMO to key muscles impacted by the disease, such as skeletal and cardiac muscle, as well as the CNS. Unfortunately, the only currently approved exon 51 skip therapy in the US results in very limited increases in dystrophin protein, about 0.3% after 6 months of treatment with eteplirsen. Next slide, please. Now, the quality of dystrophin is also critical in order to have functional benefit. I'll try to go through this at a high level, but happy to answer questions later. So here we see a milder clinical individual in BMD, in Becker muscular dystrophy individuals, who carry mutations that produce dystrophin similar to the skipped dystrophin that would result from exon skipping of similar mutations in people living with DMD. So they're producing the dystrophin as if a person with Duchenne had an exon skipping drug. So what we see here is dystrophin from exon skipping strategies may have functional benefits and lead to improved clinical phenotype. Now, I'm not suggesting that we're at all saying DMD will be converted to BMD. That's not the point. But this is just data that helps us understand that the quality of dystrophin that's produced with exon skipping will have clinical impact. Next slide, please. And in contrast to exon skipped produced dystrophin, gene therapies, as you've heard for a long time now, produces a much shorter protein called microdystrophin, which lacks many of the functional domains of a full-length dystrophin. So you can see the full-length dystrophin on the top here, and you can see on the bottom 3 rows different kinds of gene therapy constructs. And there are several different microdystrophin constructs. One that is approved, others are under investigation. And so they are missing many of those functional aspects of full-length dystrophin. And as also data on the durability of effect with microdystrophin gene therapy are still emerging, and it remains to be seen if long-term functionality is maintained. Next slide, please. Lastly, broad and effective distribution of treatments that generate dystrophin to key muscles is also important for optimal clinical outcomes in Duchenne. Not only is skeletal muscle affected in DMD, cardiorespiratory complications are the primary cause of mortality in individuals living with Duchenne. And therefore, therapeutic approaches require broad distribution to cardiac and respiratory tissues as well. Moreover, approximately 50% of individuals with DMD experience neuropsychiatric symptoms. Therefore, the ability to cross the blood-brain barrier and deliver exon skipping therapies to the CNS would be an important attribute of an effective Duchenne therapy. Next slide, please. So Dyne 251 is currently being studied in the phase 1/2 DELIVER trial in ambulant and non-ambulant males with Duchenne who are ages 4 to 16 and have mutations amenable to exon 51 skipping therapy. A total of 54 participants have received Dyne 251 across 7 doses and dose regimen cohorts in the placebo-controlled MAD portion or multiple ascending dose portion of DELIVER. That's the first 6 months of the DELIVER trial. I'm pleased to say the MAD portion is now fully enrolled. And I will also just point out that the doses you see here on the staircase refer to the PMO component of Dyne 251. Now, participants treated during the MAD portion of the study had the opportunity to transition to 20 mg per kg PMO equivalent dose in the open label extension and the long-term extension phases. As you see, we did have a 40 mg per kg cohort. So participants who were randomized to the 40 mg per kg Dyne 251 arm have all transitioned to the 20 mg per kg dose in the placebo-controlled period following review of the safety profile at the 40 mg per kg dose. For other participants, the transition to 20 mg per kg dose occurred at different times during the OLE and LTE portions. But now all patients are on 20 mg per kg. In this study, the primary endpoints are safety, tolerability, and change from baseline in dystrophin levels as measured by Western blot. We're also looking at secondary endpoints, many of which you are very familiar with, that include measures of muscle function like the North Star Ambulatory Assessment, the NSAA, time to rise, as well as something called SV95C, or stride velocity 95th centile. And I'll talk a bit more about that in a few minutes. I will point out SV95C has been accepted as a primary endpoint for Duchenne trials by the European regulatory authorities. And it's currently leveraged across different Duchenne trials that are ongoing or have occurred already. Now, while safety data for all participants in the MAD non-registrational cohorts will be shown here, I'll be focused on the efficacy data from the 10 and 20 mg per kg cohorts. As DELIVER was designed to be a potentially registrational study, data collected during the MAD portion of the study led to the selection of the 20 mg per kg dosed every 4 weeks as the dose and dose regimen for the go-forward registrational cohort of DELIVER. So really, in other words, we executed the portion here on the left-hand side of the slide, and we achieved what we wanted to achieve, which is a selection of our go-forward dose that we believe is efficacious and safe. And we are running that registrational cohort now. Next slide, please. Just to elaborate on that, in this registrational expansion cohort, 32 participants were randomized at 3-to-1, meaning 3 patients receiving active drug, 1 patient receiving placebo. And they are receiving 20 milligrams per kilogram of PMO dosed every 4 weeks of Dyne 251 or placebo. I'm pleased to say the cohort is now fully enrolled with data planned for late 2025. The primary endpoint is change from baseline in dystrophin protein levels as measured by Western blot at 6 months. And we have a number of additional biomarkers and functional endpoints also being studied, such as NSAA, stride velocity, and time to rise. Next slide, please. So these are the baseline characteristics for DELIVER, and I won't go through this table in detail. I'll point out a few things. First, the cohorts are generally similar with a few notable exceptions. The 20 mg per kg cohort is on average older with an average worse function at baseline. And you can see this is reflected, for example, in the lower NSAA score at baseline, a slower 10-meter run walk, and slower SV95C. Therefore, participants in this cohort have more severe disease relative to the 10 mg per kg cohort. Now, based on the NSAA 10-meter walk run and SV95C measures, baseline TTR or time to rise may appear as an outlier between those two cohorts. However, participants who could not complete the time to rise were excluded from the analysis, and missing times from these participants were not imputed so that we could adjust for that. Next slide, please. Now, we firmly believe the true goal of therapies for Duchenne should be to improve function. Dyne 251 resulted in long-term improvements versus baseline across multiple functional endpoints, including the NSAA, which is on the left-hand side, the time to rise in the middle, and 10-meter walk run time, which is on the right-hand side. Now, in the 10 mg per kg cohort, as you can see in the sort of the teal line, there is continued and consistent benefit across these functional measures through week 73 or 18 months. As mentioned before, upon entering the open label extension, participants on the 10 mg per kg dose were non-uniformly escalated to 20 mg per kg, with all participants at the higher dose at the month 12 assessment. So month 12, they're all on 20 mg per kg. Therefore, the sustained effect primarily reflects treatment at the 20 mg per kg Q4 weekly or every 4 weeks regimen, which is indeed our selected go-forward dose for the registrational cohort in DELIVER. Now, extension period data for this cohort show the benefit of higher dose and deepening effect on function with longer time on therapy. You can also see improvement or stabilization versus baseline are also consistently seen at 6 through 12 months in the 20 mg per kg cohort, which is in this dark blue line. Now, this is especially notable given that this cohort was more functionally impaired at baseline, if you recall, in terms of what I said earlier. Next slide. Now, an additional functional assessment in the DELIVER study is the stride velocity 95th centile. It's a digital and objective continuous measure of ambulation in the patient's normal daily settings. So this is a continuous measure of function in a real-world setting. It has been shown to correlate with the traditional ambulatory measures such as NSAA and 6-meter walk test. And it's been validated as a primary endpoint in Duchenne clinical studies by the European Regulatory Authority. Now, SV95C demonstrated greater sensitivity, meaning that it can detect change earlier versus other motor function tests. And the proposed MCID, or minimally clinically important difference. So the proposed minimally important difference in using this measure is 0.1 meters per second. I'll ask you to keep that in mind for a slide coming up. Now, very importantly, this is the way that this was developed in terms of this minimally clinically important difference is through something called a distribution-based threshold and corresponds to about a 30-meter change on the 6-minute walk test, which is known to be clinically meaningful. Next slide, please. So Dyne 251 led to early and long-term improvements in SV95C at both the 10 and the 20 mg per kg dose. Now, improvements versus baseline are sustained through 12 months for the 20 mg per kg cohort and through 18 months for the cohort that was initiated at 10 mg per kg, and then everybody's on 20 mg per kg at 12 months. Now, as with the other functional data outcomes, participants in the 10 mg per kg cohort transitioned to that 20 mg per kg in a non-uniform way. Everybody's on 20 mg per kg at 12 months, as I said. Therefore, the sustained SV95C effect primarily reflects treatment at that 20 mg per kg every 4-week dosing regimen. Now, importantly, change from baseline in SV95C following treatment with Dyne 251 achieved the proposed MCID by 6 months, thereby reflecting early and meaningful functional benefit of treatment with Dyne 251. So we believe the totality of functional data shows that treatment with Dyne 251 consistently results in early and sustained benefit across multiple motor function assessments and across different levels of baseline function, different levels of where these individuals start when they enter the study. Next slide, please. So lastly, the functional benefit of Dyne 251 is it's underpinned by the dystrophin produced. This slide shows dystrophin analysis as measured by Western blot for both muscle content unadjusted and muscle content adjusted dystrophin. So at the 20 mg per kg dose, Dyne 251 resulted in about 3.7% of dystrophin. Now, when we adjusted for muscle content, and you can see how we did it here, we wanted to be transparent and we put the calculation on the slide. When we calculated when we calculated the adjusted dystrophin using this formula, Dyne 251 resulted in 8.7% dystrophin. The KOLs felt it's very important. They guided us to make sure that we put both numbers out to the community so that this can be properly interpreted. So this robust production of dystrophin is really driving the functional changes that I described earlier. Next slide, please. So there's a lot of information. I'm going to maybe just hit the highlights. Overall, Dyne 251 demonstrated a continued favorable safety profile, with the majority of treatment-emergent adverse events, or TEAEs, reported as mild or moderate. Substantial safety data has been collected in the liver, with total doses of administered study drug approaching 1,000 over 77 patient years of follow-up, with some patients on study for nearly 2.5 years. These data also reflect over 500 doses of study drug administered at the registrational go-forward dose of 20 mg per kg. There were 3 serious TEAEs potentially related to study drug that occurred in 2 participants that were in the 40 mg per kg Q4 weekly cohort. Now, these have been previously reported. We've seen nothing since then, and both participants have fully recovered. As I said, there have been no other safety signals of note. Outside of the 2 participants with related SAEs, no other participants had persistent anemia or thrombocytopenia. There have also been no clinically meaningful electrolyte changes, including low magnesium or potassium, as has been seen in other programs. Next slide, please. Now, this data set from DELIVER illustrates the promise of the FORCE platform in developing next-generation exon-skipping therapies and support the DMD pipeline expansion that we are planning. So we believe there's an opportunity to leverage our platform to address additional mutations. Preclinical programs targeting additional exons include 53, 45, and 44, and these programs are moving forward. Next slide, please. So just to summarize, this is a lot of information that I know I've presented. The FORCE platform has achieved broad distribution to key muscles, such as cardiac and skeletal muscles, as well as CNS, in the preclinical models that we've used. The phase 1/2 DELIVER study is an ongoing randomized placebo-controlled trial in both ambulant and non-ambulant males with DMD that have mutations amenable to exon 51 skipping therapy. The safety profile is favorable to date, in our opinion, and we've had robust expression of near full-length dystrophin following treatment of Dyne 251. We're also really excited to see the early and sustained benefit of treatment with Dyne 251 that is consistent on both clinical and real-world functional outcomes out to 18 months on various measures. Importantly, improvement versus baseline in the SV95C is a very reliable measure of continuous real-world function, and we've demonstrated this at the registrational dose level of 20 mg per kg. The registrational cohort is now fully enrolled, and we do have data planned for late 2025, and we continue to move our pipeline forward. Next slide, please. Now I'm going to just switch over to an area that really hasn't been discussed in great length over the last few years. And really the point is that one critical aspect of the multi-system nature of Duchenne that, again, feels like it's been overlooked for a long time in research is the brain involvement in DMD. And of recent, thanks to the amazing work of the BIND Consortium and other efforts, much more research is being done and published in this area. There has been recognition of some core areas of brain involvement identified as particularly affecting individuals with Duchenne. So these include neurocognitive issues, where people often experience mild to moderate intellectual disabilities or particular challenges in verbal or working memory, executive functioning. Another area is academic and learning challenges are thought to be common, especially in reading, dyslexia-like symptoms, and arithmetic, maybe difficulty with numerical reasoning and problem-solving skills, and this can impact school performance. There's also increased risk of anxiety and depression that's often observed, and it's often linked to social difficulties, frustration with physical limitations, and progressive loss of independence. And finally, neuropsychiatric issues are commonly seen. And this includes higher prevalence of autism spectrum disorder, ADHD, OCD, likely due to dystrophin-related brain dysfunction that can affect some of the synapses in the brain and how the neurons are connected to one another. Next slide, please. So we wanted to investigate this with the FORCE platform. So this slide shows that the FORCE platform delivers our exon skipping PMO and induces exon skipping in the brain of a DMD mouse. Given the pervasiveness of neurocognitive manifestations in individuals with Duchenne and knowing that the TFR1 is expressed in the brain, we wanted to test if the FORCE platform can deliver payload to the CNS to potentially address brain symptoms of Duchenne. So what we used to study this is what we call an HTFR1 MDX mouse model. So this is the MDX mouse model that you're all familiar with, except it expresses the human transferrin receptor, which allows us to test our conjugate. Now, here in the top row, you can see that 7 days after administration, a single intravenous dose, IV dose, of our tool compound designed for this mouse that has a mutation of exon 23. So an IV dose of FORCE M23D, but not the unconjugated M23D or a non-TFR1 targeting control, led to widespread PMO distribution to the CNS, including key areas like the prefrontal cortex, the hippocampus, which is known to have a role in learning and memory, for example, and the cerebellum. The bottom row graphs show that this widespread PMO distribution in the CNS resulted in target engagement as demonstrated by exon 23 skipping. So we're pleased to see distribution and exon skipping in the brain. Next slide, please. So the effective PMO delivery and exon skipping were maintained at 28 days after administration of a single FORCE M23D dose in these different brain regions, in the cortex, in the cerebellum, as well as the deep brain. And the deep brain is a catch-all term for the anatomical region that's likely to contain very small features of the brain, like the amygdala. Also, this effective PMO delivery and exon skipping in the CNS restored expression of dystrophin as seen in the graph on the right-hand side of the slide. Next slide, please. So I'll talk while this video is playing, but please look at the video. Well, what about the impact of the FORCE M23D CNS delivery on phenotype? Now, it's known that the MDX mice exhibit anxiety-induced freezing behavior in response to restraint. So if you squeeze their neck and they sort of have this freezing behavior. And so this behavior is also seen in the MDX mouse that expresses the human transferrin receptor. And so you can see it in the top of the video. Restraint-induced freezing was assessed by grabbing the mice by the skin of their neck for about 15 or 20 seconds. And this causes the mouse to freeze, as you can see on the top. Now, this video is playing on a loop. So the video is moving, but you can see that the mouse is frozen on the top. The bottom panel on the left-hand side shows what happens in the presence of force. And now you can see in the presence of force on the bottom left that this mouse is moving. The graph on the right shows that a single dose of force can lead to a complete and durable resolution of up to 2 months post-dose of this anxiety-induced freezing behavior in the HTFR1 MDX mouse. So we're eager to try and figure out in the clinic what the effect of the Dyne 251 conjugate is. Next slide, please. So maybe just a few takeaways. First, the force platform leverages the body's natural cell biology, the natural expression of the TFR1 on muscle cells and cardiac muscle that allows targeted delivery of our exon PMO, exon skipping PMO, to the nucleus of the muscles where it needs to go. And we achieve broad distribution as well, which is really important. Dyne 251 has shown to be have a favorable safety profile with some participants on therapy for 2.5 years and has shown long-term improvement in measures of clinical and real-world functional outcomes. And we believe the FORCE platform has the potential to transform the lives of individuals living with Duchenne and other neuromuscular disorders by addressing the totality of symptoms experienced by these individuals, including the CNS manifestations. Next slide, please. So with that, I would like to, again, thank Cure Duchenne for the opportunity to present to you today. And thank you to the families, investigators, and sites, which you can see on this map here, that have made this and so many other advancements in Duchenne possible with all of your countless sacrifices and your dedication. What you have all done over the years is nothing short of extraordinary. And thank you so much on behalf of Dyne.
Ash, thank you. That was quite a tour de force and a lot of information to absorb. Let me invite Catherine and my colleague, Dr. Liana Orlando, our Vice President of Research, to join me for the Q&A session. Welcome, guys. So let me start with an easy one to begin with. Obviously, the community's always interested in where you are today, but thinking ahead, because there's many patients that I know that are on the call that are thinking about other exons. What's your timing for some of these follow-on exons?
Yeah, great question. So, as you know, we have to have discussions with regulatory authorities as we plan our steps forward for the other exons. We are moving forward on 53 and 45 and 44, as I mentioned before. Those are in preclinical stages. So as we get more information and more clarity on timing to get into the clinic and even beyond that, we'd love to be able to come back to the community and talk about it a bit more. I will add, and I'm sure Catherine will want to jump in here too, that I think it's also going to be very important from a regulatory point of view that companies like Dyne and others hopefully can leverage the platform technologies guidance that the FDA has produced. I know that Cure Duchenne has also been quite vocal in their perspective on that guidance as well. And so that's a real opportunity to be able to move additional exons, hopefully more quickly through the regulatory pathway. So we really need to partner with the community and with regulatory agencies to be able to move other exons forward efficiently. Catherine, do you want to add anything?
Nope, just a huge thank you to Cure Duchenne because I know that during the open comment period, the organization submitted comments when they were open for the platform technology designation. So thank you very much for that. And as Ash said, I think, we can wait and see if that will provide the much needed efficiencies, right, in the drug development process to move additional exons forward.
Yeah, thank you for that, Catherine. And I totally agree. I think it's going to be an important part of the company's strategy as you start to build out and fill the pipeline. Keeping it in a sort of regulatory environment for the minute, I'm delighted to see the SV95C as part of the trial. And obviously, it's demonstrated greater sensitivity to change over time, which is why it's a really important endpoint in Europe for approval. Do you get a sense that as you're collecting that data, there's an opportunity here to discuss with the FDA in future that this should be an important consideration for them?
So yeah, we certainly believe that there is a valid discussion to have with regulatory agencies beyond Europe on the SV95C. It has gone through a tremendous amount of validation work. It is a sensitive, reliable, reproducible measure, as been shown by published literature. And so we're really excited to be able to move our program forward, have discussions with the FDA and other regulatory agencies about using SV95C in our study. And so absolutely, I think there's discussions we'll have, and there should be an openness to it.
Okay, thank you for that. The data that you showed, Ash, is consistent and extremely encouraging. We're seeing these improvements in multiple functions, which are really starting to tell a real story about how the drug's behaving with North Star, time to rise, etc. At what point in the future is the company thinking about helping patients make decisions about switching from one exon 51 skipping therapy to another, to Dyne, for example? Because I could see that the data there is so compelling, for patients that are out there.
Yeah, I think that's a really, really good question. So, I think that the wonderful thing is that there are just more and more choices for families in the US and starting to be more for families outside of the US. Our data set, as you know, the inclusion-exclusion criteria did allow individuals who are on prior exon skipping therapy. There was a washout period of 12 weeks, and then they can go on to Dyne 251. So, our overall data set, not surprisingly, will contain individuals that washed out of eteplirsen, for example, and went on to Dyne 251. So our totality of data set that we'll be able to provide regulators and eventually, to physicians, will include some of that data. So we're excited. I will say that, and not, I just want to be really clear. Any decisions around switching or the like, obviously families have to talk to their physicians about what's best for their families. But we're excited. We have monthly dosing. I think our data is incredibly robust, is producing dystrophin and functional benefits well beyond standard of care. So we're excited about providing this option to families.
If I could jump in, thank you for that question. I'll just jump in because it gets to one of the questions that came in. And so since we're talking on this topic, that, the FDA approved drug, skipping exon 51, makes 0.3% of dystrophin. And depending on whether you're muscle adjusting or not, Dyne 251 so far has been showing between 3.5 to 8.7%. So that is, a significant amount more. So that's part of the information, right? Which would maybe help individuals consider options.
And may I add as well, because I think, Mike, part of your question to Dyne is also, kind of answerable in terms of contributions by Cure Duchenne, because I think if the community has, options for approved therapies, right? I mean, there's certainly an opportunity for Dyne and any company to partner with your organization to really provide the education, whether it is scientific education or if it's language that's needed to enable the conversation at the level of the provider, right? I think that's something that we would be, very eager, willing to work with you all on.
That's great.
Excellent, thank you. Can I just tease this out a little bit further, just to try and get a sense of where the company's thinking? Right now, the muscle biopsies are showing significant dystrophin levels at 6 months. Right. Is there plans to look at that there at a later time point, Ash, and to look for accumulation, given the half-life of this protein is very, very long?
Yeah, no, it's a great question. We talk a lot about it. Families ask us as well. So within the now the registrational cohort, it is the plan is to have biopsies at baseline and 6 months. We have really tried to listen to the community, and the burden of these open biopsies is significant. So we're taking open biopsies at the level of the bicep, and that's two. To go in a third time, I think, feedback we've gotten is that's quite a big ask. So we are not going to be looking at dystrophin levels at later time points as part of the current protocol. And again, what I'll anchor on is functional benefit.
Mhm.
To your point, right? There is clear preclinical, but also clinical evidence that dystrophin accumulates over time. And even with eteplirsen, after 180 weeks, there's about 1% dystrophin. So it's pretty small, but it's accumulated from 0.3, over 180 weeks to something that's still, quite low. So we fully understand from both preclinical and clinical evidence that dystrophin, to your point, accumulates over time. And so, perhaps it's reaching steady state at a year or a year and a half, and dystrophin levels should be much higher than the 3.7% unadjusted that you're seeing. So we fully expect that to occur because that is the biology that we see with other data that's been published. But we are not planning on burdening families with additional biopsies to look at later time points as required in terms of any requirements as part of the protocol.
Okay, thank you. That's quite clear. Also, in the non-clinical world, we've seen quite a lot of cardiac dystrophin restoration with this approach in mice. And for me, it will be interesting to continue to follow some of the older boys over many years to look for the cardiac benefits that you're seeing in there. And I know that there will be part and parcel of your thinking, as you start to look at this in the real world environment.
Yeah. No, I completely agree. Within Deliver, we are looking at pulmonary function. We are also looking at cardiac function, for example, in the form of cardiac MRI.
Mm-hmm.
So to your point, I mean, we're fully there with you. We think it's really important to be looking at cardiopulmonary effects over a long period of time. So we have it in the Deliver study, and so that we'll have that data, and then of course we'll want to follow, especially the older boys over a long period of time to be able to understand the benefit. And the nice thing too is there's some decent natural history study against which to compare long-term cardiac and pulmonary outcomes as well.
Yeah, excellent. I knew you guys would have plans about that. One other thing
Building
before,
upon that
go ahead,
theme,
Leanna.
I was wondering also, are there, since you mentioned the sort of neurocognitive aspects, are there even exploratory measures that you're doing in Deliver or any of the long-term extensions to look at changes in CNS aspects?
Yeah, it's a great point. So, we're really have been trying to characterize the CNS effects with our conjugate in those preclinical models that I was mentioning. I'll answer your question in a second. I'll get there. But within our myotonic program, we did include a measure that allows us to look at CNS effects, such as fatigue, sleepiness, cognition, communication. And we've shown benefits in individuals with myotonic dystrophy type 1 on those aspects within that program. Within Deliver, we haven't had those instruments or such instruments, and we are still just trying to characterize we're trying to understand what exactly we should be measuring that's specific for individuals living with Duchenne. We've had great advice from luminaries such as Professor Montoni and others who've been part of the BIND consortium or leading the BIND consortium. So neurologists, neuropsychologists, psychiatrists helping us understand what we should be evaluating. So we do plan to be exploring that in the phase 3 study.
Thank you. We've got quite a few questions on that, so it was good to get that update as well.
Do you want to grab a few of those questions from online?
Yeah, so there's a couple of questions too about combinations of whether or not this can be used. Certainly, I think individuals, I think in your trials, right, have been on steroids. There's some questions here about other combinations of therapies like givinostat, or even potentially in the future looking at this in individuals who received gene therapy like Elevidys.
Sure. So this is, I think, a very hot topic that everyone is talking about, rightfully so. So Deliver is a phase 1/2 study, and as as the community knows, you have to take a bit of homogeneous sort of approach. You want to approach phase 1/2 studies with a relatively homogeneous population to really understand the benefits of your specific compound that's being studied from an efficacy point of view and from a safety point of view. So for the phase 1/2 study, we've not allowed individuals who are on prior gene therapy or givinostat. We do allow those individuals who are on vamorolone, because we require patients to be on steroids. So we within Deliver, there is no exploration of combination therapies. Now, moving forward within our overall plans to understand this drug, we will be thinking about how to explore different combinations. We're not there yet in terms of exactly what we're going to do. And when we have more definitive path forward on that or plans on that, I'm happy to come back to this community and talk about it. But we understand there is a desire to understand exon skipping drugs with gene therapy, exon skipping drugs with givinostat, or even givinostat, exon skipping, and gene therapy. And so I'm sure we, like many other companies, are thinking about how to do this and the right timing for this. I don't know, Catherine, if you have anything else to add on that. Okay.
Okay, we've got some other, these are even more forward thinking. So, but whether or not this technology can be applied to address duplications or skipping multiple exons at once.
Yeah.
And so I recognize that these aren't things that are, very near to being in the clinic. This is more of a thought experiment, but if you could just at least speak to that.
Yeah, it's a good question. So I'll admit, first of all, folks like Professor Flanagan and Montoni are probably definitely far more knowledgeable about this. What I would say is that theoretically, it is possible for exon skipping drugs to be used for duplications if you're able to skip. So if you have a duplication of I don't know, exon 51, theoretically, would an exon skipping therapy be useful if you can skip one of the exon 51s and not both to produce full-length dystrophin? I think theoretically that's on the table. But I think there's a lot of work that needs to be done to be able to understand how this works. I will point to some recent work that was done by Dr. Flanagan, in which he looked at duplication, DUP2, boys with DUP2, and provided and delivered an exon skipping drug within a gene therapy, within a capsid, within a gene therapy construct, and delivered it and was able to show some positive effects treating these DUP2 patients. So I think it's theoretically possible. A lot more work has to be done, and there's people who are far more knowledgeable than I am. In terms of skipping multiple exons, again, I think there's theoretically that is possible. We obviously have a payload or an ASO that is specific to skipping exon 51. Could there be technology that's developed where you can attach an exon 51 skipping drug and an exon 53 or whatever to skip multiple exons? Again, I think theoretically that's possible, but there's so much more work that needs to be done, and people far more knowledgeable than I, such as Mike Kelly.
Yeah, the thing that I would add in there, Doug, just to help you out, is that
Thank
one
you.
of the keys to actually skipping multiple exons is going to be the efficiency of skip. So in the old world where we had very inefficient exon skippers, it made it quite challenging to skip multiple exons. I think now with technology like the Force platform, it opens up that door where we can now test and ask those questions. And I think that there's going to be something that will be done over the next months and years.
Yeah. Yeah. And you know, I'll add even, Liana, right? There's also intronic mutations, you know, that act, right, like deletions. So theoretically, exon skipping drugs could be used for intronic mutation. So I think there's a lot of great thinking out there that has occurred and more work that has to be done.
No, and I think, thank you. And I think the things that you said before in terms of, you know, even your next single exon skipping programs, right? Anything that we can leverage as a community, you guys as a company, in terms of once the platform has gotten over the finish line the first time, being able to lower the criteria for actually tackling all these follow-on things will just help us get to, you know, more and more cases of unmet need.
Okay, is there any final question you might want to pick out?
I think there's an important question in the list of great questions, is the question around if you increase the frequency of dosing, could patients have that option, thereby increasing the amount of dystrophin? Say every 2 weeks rather than every month or every 8 weeks.
Yeah. No, I think it's a really important question. And not that we have time to get into it, but we have done a tremendous amount of preclinical work in both in mice and in NHPs to land on what we believe is the optimal dose regimen that balances efficacy as well as provides a very wide therapeutic index or a very wide safety window. And so, again, as we've always been guided to by KOLs, this is a lifelong condition. And so think of, you know, treatment in a rational way. And so we know that dystrophin will accumulate over time. And so we feel like this anchoring on every 4 weeks, given for, you know, the lifetime of the individual, will provide dystrophin levels that hopefully will be transformative. And so going to every week or every 2 weeks, one is not necessary from all the work we've done. Two, it probably will not provide additional benefit over that long term. And three, it's just the burden on the families that we're trying to also be mindful of and reduce with monthly dosing.
Good points. Thank you for that. And thank you for that final question. I really appreciate it. We're almost right at the top of the hour. I want to thank everybody for joining the webinar today. And a big thank you to the whole team at Dyne Therapeutics for the work they've done developing Dyne 251 and for sharing the important update with the community. We look forward to future updates as the pivotal trial is now fully enrolled, with the potential biological licensing application submission for the US accelerated approval pathway in early 2026. A post-webinar email will be sent tomorrow should you have any additional questions. And we'll be in touch with all attendees to let you know when the webinar will be posted online for review and for sharing with families and others. So thank you again for joining us today, and have a wonderful rest of the day.
May I make one comment, Mike? So
Sure.
I'm so sorry. I don't know if we showed the last slide, but if you have any other questions for us, please reach out to Catherine. Her email can be provided to you through Cure Duchenne. So sorry about that, but thank you.
Oh, thanks. Thanks for that, guys.